I want to start by thanking the committee of MCBIOS 2020 for providing me this opportunity to present my research work. So the title of my presentation today is called Robust Partial Reference-Free Cell Composition Estimation in Tissue Expression Profiles. So the structure of my presentation is very simple. I will start with some background to motivate our project. Then I will go to the our proposed method for robust partial reference-free deconvolution, and then I will present you some of the very interesting results we obtained from simulation study and real data analysis. I will end in a very brief summary of our project and the implementation of our method. So some background. In the early days of high-throughput data analysis, people study cell line samples. But in recent years, there's a growing number of interest in indirectly collecting samples from human body. The samples collected, including tumor, brain, and blood, they are all heterogeneous samples that consist of many different cell types. So all those different cell types, they have their distinct high-throughput profiles, and they correlate with human disease in different ways. So the cell mixture complicates the data analysis in high-throughput data experiments. So take the gene expression data as an example. There are two quantities we're usually interested in learning from the sequencing of high-throughput sample. The first quantity is the average gene expression profile of different cell types from the sample. The second quantity is the estimated proportion of different cell types across all the samples. And the second object is the topic that we're interested in learning in our current project. And we also call it cell composition. So why cell composition is an important parameter to estimate? At least two major reasons here. There could be more. The first reason is that cell composition is of interest for direct use. Many studies found that the composition of immune cell types is a strong predictor for the treatment response of patients who received cancer immunotherapy. Some other studies also found that neuronal cell death is a prominent feature in Alzheimer's disease brains. So imagine that if we can estimate the cell composition accurately for the heterogeneous samples we collected from human body, we could use those to predict a treatment response or even do disease diagnosis. The second reason is that in order to appropriately analyze high-throughput data, we should adjust for cell composition in the data analysis. A study published in 2014, the two authors found that without accounting for cell composition in epigenome-wide association study, the results could be biased or even wrong. Due to the reason that cell composition is an important parameter to estimate, there have been many existing methods that were proposed to estimate cell composition from high-throughput data. I will show a motivating example first and walk through the existing method in a minute to tell you why those existing methods cannot solve the question we're interested to solve here. So our motivating example comes from data from our collaborator from Baylor College of Medicine. So in this data set, we get 9 RNA-seq data from 9 mouses. so the tissue of interest is the ovary tissue from mouse, and there are 3 groups. The second and third group of mouses, they all have Fragile X syndrome, but the third group is more severe than the second group. So our collaborator wants to understand how cell composition change in different groups of mouses. There are a few prominent features of our dataset. The first is that the sample size is very small. There's only 9 mouses in total. And due to the limited budget, we cannot do extra experimental other experiments like cell sorting experiment to understand the cell composition. The second feature is that from some prior knowledge, we know that there are 4 main cell types in mouse ovary, and we know what are the marker genes for each cell type. The marker genes means the genes that are only expressed in one cell type, but not in the others. The last feature is that there's no pure gene expression profile for each cell type, meaning that there's no reference panel for us to use in the deconvolution of those samples. So some existing methods to estimate the cell composition. The first is to use cell sorting technology, but those methods are very expensive and very laborious, thus does not applicable in our study. Then we resort to in silico computational method. This reference-based deconvolution group of method is the most widely used, but we do not have a reference panel, so we cannot use those methods. The second group is called reference-free deconvolution. We also have one method published last year in this category, but the problem with those methods is that they usually ask for a big sample size, like more than 50 or even more than 100, to get an accurate estimate. This means that reference-free deconvolution also does not applicable here. The last group of method is called partial reference-free. It does not ask for a reference panel, but it will require some external information to guide the deconvolution, which will already show that we have some marker informations. So we try this group of method on our data. The first existing method, DSA, take a very simple approach. So it uses the cell type specific marker in the deconvolution. It just takes an average of all the markers per cell type and then estimate the cell composition in just one step. We try this method in our data, but the result is it's not very good. Some of the estimated cell composition are even negative, which is illogical. The second method is called Bayesian cell count estimation, BCCE. So BCCE asks for the prior knowledge of cell proportions, but BCCE only applies for DNA methylation data. Realize that our question cannot be solved by those existing methods. We decide to propose our own method. So we call it TOAST for the analysis of heterogeneous tissue. So we hope our method will be a partial reference-free deconvolution, which can estimate the cell compositions. The input will be the data matrix of gene expression and a list of cell type specific markers and the prior knowledge of the cell proportions. But we want the prior information of the cell proportion to be optional. And then we'll be able to estimate the cell composition and the pure tissue profile. So here is a schematic overview of our proposed method. So given the gene expression of the heterogeneous sample, we use the marker information as a constraint to estimate the pure tissue profile, and then we use an iterative approach and non-negative least square algorithm to ask for the cell composition. If the prior information of proportions are available, we will incorporate such information in solving the cell composition step through a Bayesian framework. So some notations here. Y is the observed gene expression data matrix. H is the quantity that we wanted to estimate the cell composition. M is a cell type specific marker. So alpha and sigma are the prior knowledge of the proportions, which are optional. So our iterative algorithm is designed in two steps. In the first step, we estimated the marker-specific expression, and then in the second step, we solve the cell composition by non-negative least square with post hoc constraints when the prior knowledge of the proportion are not available. So if the prior knowledge are available, we can incorporate such information through a Bayesian framework. So we impose the prior distribution for the cell composition of each cell type, and then we derive a maximum likelihood-based solution, which actually have an explicit solution, making the computation very efficient. So putting our algorithm together, our method consists of two parts. So in the first part, meaning that host without prior, so if we do not have the prior information of the cell composition, we will solve the cell composition using non-negative least square. But if we have the prior information, we'll update the cell composition using a Bayesian framework, and we call it toast with prior. So we designed a series of simulation studies based on real data sets. I ignore the simulation setting details here, but in the first simulation, in the results, we're trying to compare our proposed method with two existing methods, non-negative matrix factorization, which is a reference-free deconvolution algorithm, and DSA is a popular partial reference-free deconvolution. We show that compared with two existing methods, our proposed method can achieve lower absolute mean bias and higher correlation with true proportions in our simulation data sets. So all those results are based on 100 simulation data. So I want to devote most of my time to real data analysis. So I will brief summary the conclusion from the other simulation settings. So we show that our proposed method outperforms existing method for different marker numbers and different sample sizes. Also, our proposed method are robust to noise expression and robust to the inclusion of wrong markers. And lastly, we also show that the cell type composition variation have an impact on the estimates. Larger variation in cell type composition is associated with better cell composition estimation. So I want to go more detail in my real data analysis. So in reality, there are many sources for the prior information. For example, the marker genes for blood can be obtained from microarray gene expression reference panel, or RNA-seq gene expression reference panel, or single-cell RNA-seq data, or even external marker repository. The prior information of proportions can also be get from cell sorting experiments or single cell study. So in my real data analysis, I want to show two things. So the first thing is that our deconvolution algorithm compared with existing method works well. The second thing I want to show is that how those different sources of information performs in deconvolution analysis. So we collect 5 real data analyses. The first one is a manual mix of rat tissues with known proportions, but the other 4, they all come from, say, the PBMC data or the brain sample. They are all come from real human body. We also get the single cell data from PBMC and RoseMap, and the prior proportion knowledge for PBMC is collected from an experimental study, and the prior proportion knowledge for brain is collected from AD single cell study. So in the first real data analysis, we benchmark our method with markers selected from microarray reference panel. We show that our proposed method compared with the existing method DSA have lower bias and higher proportions. But in reality, if we have the reference panel from microarray, we can directly do reference-based deconvolution. So it's more important to evaluate our method with markers selected from different platforms. This result is my favorite figure. So we evaluated how our proposed method TOAST compare with existing method DSA and CyberSort, which is a reference-based deconvolution algorithm with markers or reference panels from bulk microarray, bulk RNA-seq, single cell RNA-seq, and existing marker repository, cell marker. So this column is a marker in they are human marker from cell marker, and the last column is a mouse marker but transformed to human symbol, also from this repository cell marker. So there are 2 observations I want to point out here. The first is that compared with existing method, our proposed method always achieves the lowest bias and the highest proportion in estimating the cell composition from the 2 datasets. The second thing we found that is that the markers estimated from bulk data, like from microarray and RNA-seq experiment, are better than the marker selected using single cell data, and even better than the existing marker repositories. So the mouse marker to human symbol from existing repository had the lowest performance comparing with the markers from other resources. Here, where I'm trying to show the comparison of the true proportion versus the estimated proportion for our proposed method and existing method. The first row is the marker selected from bulk RNA-seq, and the second row is the marker from single cell RNA-seq. And this results is that how the proportion change in 4 brain regions in control asymptotic AD and AD, meaning Alzheimer's disease. So some existing study have found that AD will have an impact on the first 3 brain regions, but not on the last region. So very interestingly, we find that compared with controls, AD have lower neuron proportion in the first 3 regions, but not in the last region. But for this data set, we do not have a true proportion to compare with, but we find the conclusion is kind of consistent with the knowledge we have, and I find the results very, very interesting. So a brief summary. So here we propose a method in the category of partial reference-free deconvolution. Our method is very flexible and robust with small sample size and the noise. We implement our method in a bioconductor package called TOAST, and our method has superior computational performance for moderate data that we usually have, like say 4 cell type, and each cell type have 20 markers. Our method, for both our method, it takes less than one second to complete the computation on a regular laptop. So in the motivating example, our method have been applied to that data set, and that study was prepared for submission. Our method was accepted in bioinformatics just in March this year. And I want to finally give my acknowledgement to my advisor and to my collaborators, and I want to thank for the support from the MCBIOS committee 2020, and I want to thank you for your attention.